The human genome is defining who we are as individuals and what we will look like as we develop as humans. The unique DNA in each of us develops the cells, which we consist of, and each cell has its own purpose and structure. But how is genome folding for particular cell types, also known as neurons? That is what I'll be investigating in my project. Good morning. Neurons have to live a lifetime, which means that their genome must be organized stably and faithfully enough to last that long. But at the same time, dynamic enough to allow access to all the important genes that are needed. In our long-term vision, we want to understand how this folding is affected by mutations that so often occur in diseases. The main purpose of my research is the generation of knowledge of how cells are regulated and disease states are manifested. There are many mutations that are associated with certain diseases, but it's not clear which of them are really relevant. So this, in fact, is a very simple problem. How can the structure of a genome have such fundamental effects? If we can understand their role in the genome better, we can better predict if a patient will develop a certain disease. We know that the 3D genome is different for different cell types, and we are aiming to understand all that in more detail. Also, how and why neuronal diseases only develop at certain ages. One explanation is that the organization of the genome erodes over time, which is the focus of our work. I'm personally very interested in the mechanistic details of how things work. And this is a very complex problem that nature gave us to solve. Our daily work is to think and develop new tools to extract this hidden information. Super cool. This way, we can discover things that nobody on this earth has ever discovered before.